Hey everyone, we are the Advancing Vaccine Research through Sequence Analysis team. Thank you all for being here today, and let's get started. So, my name is Jaren, and I'm a rising junior at NC State University.
Hi, my name is Sahani. I am a rising junior at Duke University.
Hello, everyone. My name is Johan. I'm a rising junior at Duke University.
Hi, I'm Manya, rising sophomore at Duke.
And our team leads are Mark McCahill and David Lopes. Our stakeholders are Scott Schmidtler and Kevin Wehe, and our departments that
Thousands of antibody sequences, all while trying to track how diseases mutate in order to develop vaccines. That's the challenge researchers and immunologists face when studying how B cells evolve. They work with complex lineage trees that are honestly often messy, hard to read, and based on raw data that they have to interpret and then create manually on their computers. This ends up becoming extremely exhausting, time-consuming, and very easy to get lost in. So for our project, our goal was very simple, and it was to make that process clearer, faster, and more intuitive so that researchers can simply input their data and instantly generate a lineage tree that's interactive, organized, and easy to explore.
Okay, so now that you've seen what kind of challenge researchers face, I quickly want to talk about the science behind our project so that everything makes more sense as we move forward. So, when your body detects a virus, it activates your B cells, which is a type of white blood cell that produces antibodies. And as you can see on the right, this is what an antibody looks like. These antibodies are proteins made up of two chains, a heavy chain and a light chain. The heavy chain is the longer part in the green, and the light chain is the shorter part in gray. Their main job is to essentially recognize and bind to specific parts of a virus called antigens. And once they bind, they either block the virus from entering your cells or they signal other parts of your immune system to come in and destroy the virus. So, what makes this system really powerful is that B cells don't just produce one version and stop there. They go through something called somatic hypermutation, where they intentionally introduce changes to their DNA to create slightly different versions of antibodies. And some of those changes actually improve how well the antibody can recognize and fight the virus. So over time, the immune system gets to keep the ones that work the best, and that's how it builds a stronger defense. So, to understand how antibodies change over time, researchers use something called BCR data, which is short for B cell receptor data. It basically shows how antibodies mutate over time as the immune system learns to fight a virus. And by doing so, it helps researchers trace the step by step changes that make antibodies more effective. So on the right, you can see an example of what we call a lineage tree. Each dot in this tree represents a specific antibody version, and the branches show how those antibodies evolve through mutations. And you'll also notice that the labels marked with H and L, those tell us whether the mutation happened in the heavy or light chain, and the different colors indicate how rare or common each mutation is, the green being for more frequent and the red being for rare. I would say that seeing a tree like this early in our project really helped us understand what researchers are working with and why having an interactive tool to explore these patterns could make their work way faster and more intuitive. So now I'll pass it on to Nahum to talk about the core features and functionalities of our tool.
In our sequence analysis workflow, one of the primary file formats we use is the FASTA file, and it's responsible for storing biological sequence data in plain text, where each entry begins with a unique label marked by a greater than symbol, followed by a string of amino acids or nucleotides. And these files are especially helpful for tracking antibody mutations across branches of a lineage tree and really annotating those trees with real sequence data. They also serve as a bridge between antibody names and their corresponding sequences, helping us better understand genetic relationships and variations. And to improve this process, one of our goals was to integrate and build upon tools like the Armadillo web server. Another file format used to represent evolutionary relationships is the Newick file, and this format uses nested parentheses to depict evolutionary trees where each node can have a label and a branch length. And these files are commonly used to define relationships and divergence between sequences and to really serve as input for tools like biophilo and free visualizations. And in a rooted tree, the structure starts from a common ancestor and branches out from there. And we use Newick files to generate lineage trees of B cell receptors, allowing us to better understand genetic variation and their evolutionary paths. And to make these relationships easier to explore, we visualize lineage trees using different layouts. Force-directed graphs are used to arrange nodes based on simulated forces to produce a more clean structure. Radical tree layout to display the tree in a circular format with the roots at the center, highlighting the branching hierarchy. And then lastly, sunburst chart layered information in concentric rings, allowing the users to really interact with complex lineage data in a more intuitive way. Now I'll pass it to Tahani.
Yeah, so now I'm just going to run through a quick demo of our homepage and our navigation pages. So as you can see here, our web platform is called VaxPath, and right when you enter the homepage, it says Visualize, Analyze, Discover, which is the workflow that we're trying to improve. And then we have a short summary of what our tool does. The user also has two options, a Get Started button that takes them to the tool, and the Learn More button that takes them to the About page. And then down here, we have a quick synopsis of VaxPath features, so some of the features that VaxPath offers. And then as we go to the About section, this About page is where we introduce our tool's mission and how it helps researchers visualize complex lineage trees with ease and clarity. We also include a brief overview of the science, explaining key terms like B cell receptors, heavy chains, and light chains, as Jaren described earlier. And then as we go back up and we click tutorial, the tutorial page simply walks users through uploading data and navigating visualization features step by step, which is perfect for first-time users. And then on the team page, we have it highlights the developers, leads, and stakeholders behind the project with roles and LinkedIn's included. And for the contact page, if you need support, have feedback, or want to collaborate, the contact page provides emails to reach our team. And then on our FAQ page, we answer common questions about supported formats, interpretation tips, and troubleshooting guidance. We also have a citation that can be used to cite our web app right here. And we also have a link to our source code. And so now I will pass it on to Johan to guide you through the visualization tool.
Yes, I'm going over some of the features of the visualization tool. So this is our homepage. So this is our homepage, and there are two ways to get to the visualization tool, either by just clicking on the two page on the top, or by clicking on the Get Started button, like what I did here. And yeah, this is taken directly to the visualization tool page. And this is the UI of our tool. So as seen on top, we can upload a Newick file and also some two text files. So with the Newick file, it would just help generate and visualize the lineage trees. And then with the two, we can upload two separate text files, which have been processed by Armadillo from Files to do the annotations. And once those files are uploaded and click on the Show Tree button, it will visualize the lineage tree here. And as see, we can also select different tree layouts, and we'll automatically generate the layout. And for us, we utilize the Cytoscape library from the Jupyter Lab, and mainly to help design the nodes and the edges of the lineage tree. And one unique feature that we implemented is that we're able to see the nodes annotation just by clicking on the nodes here. And also, if you notice, like the visualization is interactive, like we can zoom in, zoom out, move nodes, or like click nodes, for example. And then below are like all our features are for the annotations. So we have two different modes, one for selecting antibodies and for selecting mutations, as well as being able to use the drop-down menu, like what I'm doing right now, or the search box to select an antibody. And with our annotation panel below, we can see the nodes we selected is ancestral pathway, the one I just highlighted, and the mutations are collocated based on the mutation probability with the legend below. And then as seen here, we can also search the antibody using the search box. And then one feature we added is to display the annotation for the ancestry pathway. So like for this example here, we selected the I1 antibody and we can see the annotation of its ancestors. And we can also highlight the ancestral pathway of the tree by clicking on the orange button here. And the nodes we highlighted in black. And we also have an option to only display the annotation, the ancestry pathway, just by clicking the blue button. Like here, it currently displays the ancestral pathway of the I1 antibody. As well, we can just display it with different tree layouts. And we can store the tree back to the original form. And then finally here, we could display all the annotations for each node onto the annotation panel. And now, so now onto the mutation mode, we can select a mutation and we'll show the annotation of the nodes that do have that specific mutation. So like for this example here, like there's two nodes that have this specific mutation I selected. Again, I could select different type of mutation and see the nodes that have that. And just like the antibody mode, we can highlight the nodes that do have the mutation. Like here, it'll be highlighted in black. And we can also have the option to just display the nodes that contain the mutation I selected as well. Again, we restore the tree back to the original form. And yeah, this is our current visualization tool. And I'll pass on to Manya for the bar tool.
So this tool is a little bit different from the one Johan just showed you because it focuses primarily on the presence of a mutation and the actual count of mutations in a specific gene sequence. So you're going to start by uploading a NUIC file. We're going to start with the heavy chain one as an example. And in this one, you don't use any FASTA files. You also just upload a .txt file with mutation information. We'll do the corresponding heavy chain annotation file for that. And then we can choose an example mutation like A24T. You can enter any mutation in this highlighter box and hit plot tree. You'll see yellow bars, which indicate the presence of that A24T mutation. And on the right-hand side, if you can see the gene sequences, they're in line and corresponding to the bars you see on the left-hand side. And on the right-hand side, you also see numbers, which represent mutation count in each of the sequences that are lined up with it. And you can also find that information on the legend over here. On the y-axis, you can find the different variations of the gene sequence, and on the x-axis, you can find the branch length. Passing it on to Nahom now.
Yeah, as seen here, our tech stack supports this workflow end to end. We use Jupyter Lab for interactive coding and data analysis, Viola to convert our notebooks into user-friendly web apps, and lastly, GitLab for collaboration and our version control. And all of this development is done in Python, and our tools are deployed on a production virtual machine using Clockworks with system administration handled by Duke OIT. And overall, this setup really ensures our platform to remain collaborative and easy to maintain while we expand its capabilities. Now, passing it back to Manya.
I just want to take a moment to talk about future developments and things we didn't get a chance to implement in these 10 weeks. The number one thing we really want to work on is having a functioning browser download. I assume for a researcher, it's probably tedious to come back to our website each time they need to see a graph. So it must be much easier for them to be able to save the files that we output as PNGs or any other format if we have more features. And then Armadillo integration. And thirdly, we want to have people being able to sort by mutation count. And this would be especially helpful for that bar highlighter tool where we already display the mutation number. To just be able to sort and see less information at a time might make it a little bit easier to interpret. And lastly, being able to hover over tooltips with more information for mutations. And we want to be able to allow users to select which type of information they want to see when they hover over these tooltips, just to make things a little more comprehensible.
So thank you for listening. And if you want to explore the tool yourself or learn more about how it works, feel free to scan the QR code or visit the link on this page, which I will also paste in the chat in just a second.